Thank you, Pierre. Yeah, my name is Jake Hogan. I'm with the Molecular Systems Group at the Blue Brain Project. And it is my honor to welcome our three distinguished guest speakers for the next session, Professors L. Felipe Barros, Marja-Leena Linne, and Johanna Beyer. First up is Professor Felipe Barros, who describes himself as being mystified by the uncanny ability of cells to keep themselves alive. Felipe joins us from Centro de Estudios Científicos Valdivia in the beautiful Patagonia region of Chile, where his group is developing optical techniques to characterize metabolism with improved spatiotemporal resolution, including the first genetically encoded FRET sensors for lactate, called laconic, and pyruvate, called pyronic. It is therefore not ironic that Felipe will now tell us about how you need to have glucose for making lactate. Felipe?
Thank you, Jay, for that nice presentation. And thank you, Pierre, for inviting me to this meeting. When you start to talk about brain metabolism, you start with the beginning. And this slide is probably the beginning for many talks for many people. When we saw this cartoon many years ago, it's so daring, so elegant, and so devoid of evidence in the very moment it was proposed. It was so bold of these guys to jump into the world, and they received the treatment they deserved. No, Pierre, when you are really original, you really suffer at the beginning. So over the years, we've been trying to look at this hypothesis experimentally, to probe it, to find in which sense it was right, it was wrong, and I can advance you the conclusion is that it's mostly right. Of course, it has become more complex because we know much more about the molecules involved, about the mechanisms, about maybe different neurons, different astrocytes. This is still evolving, and it's going to be a long way. I think we are midway through to really understand what it means, the coupling between neuronal activity here on the left, astrocytes in the middle, and blood supply, etc., on the right. And one of the important things that we might discuss later on with the panel is that this relationship is bidirectional. You have information flowing in both directions. A very important pivotal part of the model is lactate, of course. And it's not easy to follow lactate because lactate is like almost like a gas in the sense that it moves away very quickly, is made very quickly, is used very quickly. So difficult to really do metabolism with lactate. So in the 80s and 90s, most of what we knew about metabolism in the brain had to do with the results of isotopic studies. And lactate is very bad for isotope. If you apply lactate, label lactate, it disappears in a second as CO2 and it's gone. It's not like a glucose that you can label with deoxyglucose and get an idea of flux. With lactate, it's very difficult. I will tell you a little bit about, Pierre advanced this already, thank you, Pierre, about the mechanisms whereby lactate is released from astrocytes, which at the beginning we thought they were going to be MCTs, monocarboxylate transporters, but now we know there is an additional player that I think is going to be very exciting in the future too. And of course, lactate is made of glucose by glycolysis. So I will tell you about ways to study glycolysis in single cells and in vivo and in vitro, and again to follow glucose, which is the original stuff. And glucose comes, of course, through the blood, through transporters. And this is the title of the talk. I will tell you a little bit about new research regarding how glucose enters the cells and how is this affected by neuronal activity through potassium mainly, and also how it goes out of the cell. The mechanisms involved have we have done kind of advancing on that front is still quite murky. One of the problems with this kind of research is that the links between signaling and metabolisms are still not there. We don't really know how these things talk, but the sodium potassium ATPase continues to be a major player. And there are more signals. In addition to glutamate, I will be talking mostly about potassium, and I will not have time today for the benefit of time to talk about ammonium or nitric oxide. The way we see this now is that these signals are complementary. They travel different in space and time. They probably affect different cells in different moments. They're released by different compartments of neurons. For instance, glutamate is presynaptic, potassium is postsynaptic, and that makes a big difference. So techniques. Techniques are very important at the time of drawing conclusions, and we are jailed by them. So what do we have right now to approach these questions experimentally? We have the isotopes here on the left, which have evolved. This is a wonderful work by, this is Bombento, yes. This is Bombento data with the highest possible resolution of 2DG that gives an idea of groups of cells, and they are getting closer to single cells. Then we have the isotopes used in modern ways in which they can give you precise fluxes of different compartments with some assumptions. Here on the left bottom, enzyme-based microsensors, which been used for 30 years now. They give you better resolution because they are smaller, but still they give you a partial idea of what's going on. Optogenetics is entering very slowly in the field, and I will be talking mostly on the right here at the bottom about genetically encoded sensors, which is a nice way to probe these metabolites and pathways in a less invasive way. Of course, you still have to express the sensors, and that is not trivial. But in mammals and also in Drosophila, people are really advancing quickly and getting very nice data. So what is the genetically encoded sensor? It's a protein. In this case, this is a laconic. The Fred lactate sensor, Alejandro San Martin built in our lab about 7 years ago, which is based on a bacterial protein LLDR, a transcription factor, coupled to two fluorescent proteins, which undergo Fred. So if the distance or the orientation between them changes when lactate binds its transcription factor, that will give you an idea of the concentration of lactate in that moment. The temporal resolution is very good. Of course, it's limited by the microscope you're using, usually in the range of seconds or sub-seconds, which is more than enough for what you need for metabolism. And spatial resolution is limited by the compartment where you send this tool. So you can get even single mitochondria. We'll show you in a second. And at this stage, there are sensors for about 10 metabolites, glucose. By Wolf Frommer's lab, we made the lactate and pyruvate, and at Yelen's lab's done ATP. There are groups in Japan that did ATP and NADH, also paired up there. There are many sensors. They're coming quickly. They're getting better every time. And more recently, calcium people have become interested in metabolism, and they are using all their knowledge acquired to perfect GCAMs to improve the metabolite sensor. So the future is bright in that sense, and we expect that in years to come, many other metabolites will be followed uh, in real time using these sensors. And of course, when you start looking at things in real time, then the models become much more powerful. So this meeting is about modeling, and we really need to feed you guys with data, powerful data, so you can make the models more predictive. And this is something that I think it will happen. So an example of the uses of these sensors, this is laconic in a culture. Uh, the blue thing is a neuron, and the red is an astrocyte. That shows you, gives you an idea of the difference between lactate concentration between these two cells in a culture. This gradient Pierre was mentioning before. This is a new version of the pyruvate sensor now expressed within mitochondria. This is Drosophila, and you can see beautifully individual mitochondria. So if you can see it, that means you can measure it. So we can measure now pyruvate or mitochondrial flux in single mitochondria, which is mind-boggling. This is very, very interesting. And this is in a picture by Bruno Weber's lab, showing in a cranial window laconic and pyrronic expressed in two areas of brains in different astrocytes and neurons. So using a microscope that can switch between the four fields, you can have lactate and pyruvate in real time in neurons and astrocytes in the same experiment, which is amazing too. And this has been instrumental to address one of the big questions of the lactate shuttle hypothesis, which was the gradient between astrocytes and neurons. Of course, you get better experimental control in vitro, but you get more physiological relevance in vivo. And it is nice to be able to combine these approaches, and then you know what you're doing. So Pierre showed already this story by Bruno's when they were able to show guys and were able to show the gradient between the two cell types. A more recent development of this story is now the time dependence of the changes in lactate concentration in neurons and astrocytes. So on the left, you have this paper by Paul Gould's group showing how a behaving rat in this case shows a lactate rise, a very fast lactate rise. This is the start of a memory task. This is a behaving animal. And if you look at it carefully, the lactate rise is before the glucose drop, which is interesting. And we may discuss later on this what it means, this temporal dissociation. What Bruno, Mark, Ayman, and Bruno and colleagues in Zurich could do was to take it to the next level and measure at the same time in extracellular space, but within astrocytes and neurons. And the result is very, very nice. So what did they found? They found repeating the electrode data that you see a rapid increase in extracellular lactate. At the same time, you see a decrease in astrocytes, and nothing happens in neurons. And about 20 seconds later, you see a rebound, increase in astrocytes, and then you see the lactate increase in neurons. So modeling this data, the most economic explanation is that you have a release of lactate from astrocytes that appears in the extracellular and then goes into the neuron. So this is the lactate shuttle in real time. In the same paper, they were able to to show that the gradient that was found in anesthetized animals is also present in awake animals, which was important. So the phenomenology is fantastic. It tells you an idea of what's going on, but mechanisms is something that we are very interested in the lab. So we wanted to know what are the mechanistic explanation for these changes. And actually, the paper appeared 5 years ago by Tamara Sotelos in the lab, found that astrocytes, they keep their lactate above equilibrium. So, which is not obvious, because if you look at the text, most texts would say that lactate transporters are so fast that everything is equilibrated, which is not true for astrocytes, even though they're producing masses of lactate, and there's lots of lactate in the extracellular space, they keep it higher. So there's a resistance in the exit. And now the most exciting thing is that when you activate these cells with potassium, a channel opens at the surface, which releases the lactate. So this is a lactate reservoir, which is mobilized on demand by neurons through potassium, which is a postsynaptic marker of activity. And this is how it looks when you see this happening in the tissue, which is the same that Bruno and co-workers saw in the awake animals later on. So when there is local stimulation, you see first this lactate dip, it's the lactate channel opening and pushing the lactate through using the membrane potential of the astrocyte, which is very negative. And then you see an overshoot, a rebound. Our interpretation of this rebound, and this is glucose being metabolized to lactate through glycolysis. So you have a sequence of the lactate, very fast lactate channel, and then glycolytic activation, which comes after. If you look at glutamate, glutamate also activates glycolysis. This was shown in that epic paper by Luc and Pierre. But the effect of glutamate is a bit slower. So this experiment shows you here now glucose concentration in real time using Wolframmer's sensor. And that slope gives an idea of the glycolytic rate before the stimulation with glutamate. And this very small slope, but about 20 minutes later, look at the slope. So this is strong activation of glycolysis. And thanks to the time resolution of this methodology, we could repeat what Pierre had shown before, but now to check the time course of the activation. And after 30 seconds, you don't see very much. 5 minutes, a little bit, and this is very strong after 20 minutes. So here, glutamate and potassium are doing different things. Potassium is very fast in the activation, which is, I'm getting ahead of myself. I didn't, I will talk about glutamate first. So glutamate needs a time for the activation. And this is important for the discussion we're going to have later, maybe regarding the role of calcium and the connection with metabolism. Potassium, on the other hand, which is a very good readout of postsynaptic activity, as is shown here on the left, it really moves a lot. This is a recent paper by Mike and Nedergaard's group, where they measure extracellular potassium in behaving animals. And look at this, this is extracellular potassium in the motor cortex. And when the animal runs, whoops, goes up to 1 millimolar. This is quite amazing for an extracellular ion. And this happens all over the brain. So it's not related with local activity only. This is probably arousal, activation of whole brain. And a bit earlier, we found that potassium is a very fast activator of glycolysis. So here we are measuring the rate of glycolysis. This is slope in steady state. And then upon increasing potassium to 3 to 6 millimolar, you see a threefold activation, and then even more when potassium goes even higher. This is instant. It happens immediately when you add the potassium. And this is dependent on one protein at the cell surface, which is the MBCE1 bicarbonate sodium cotransporter. And when you knock out this transporter, the decrease in glucose brought about by the activation of glycolysis within astrocytes reverts to an increase. So it goes to the other side. It's quite amazing. And now we know, we explain why this increase is seen. It's because the glucose transporter is also activated by potassium, probably by a mechanism which is independent of the MBCE1. And this is a long story, but I will tell it very shortly. To be able to see this, we have to develop a new method to be able to measure glucose transport in the presence of glucose, because the mechanism depends on the continuous presence of glucose. And if you look at the slope here, this is 3-OMG, 3-O-methyl glucose, which is a glucose analog. This gives you an idea of the glucose transport capacity of the cell. Upon potassium activation, there is a decrease. This is the decrease due to metabolism, but the permeability is increased. And this is, you can get the permeability data using some minimal model. And this is the dose response curve of the activation of the glucose transporter. And this has consequences. So you activate the glucose transporter and glycolysis at the same time. And what happens was, in terms of the metabolic state of the cell, was a surprise. We were expecting, because the sodium pump will be activated, the ATP levels would go down, the classic homeostatic response that we found in most texts. But astrocytes do it differently. So when you applied afferent stimulation, ATP goes up instead of going down. So this means, in our interpretation, is that glycolysis is, the activation of glycolysis is stronger than the activation of ATP consumption by the sodium pump. It's different with glutamate. With glutamate, you see the standard homeostatic response, that ATP levels go down. But with potassium, because the glycolysis is so strong, then ATP go up. And this weird reaction to a extracellular stimuli brought this idea of looking at respiration. So how a cell would react when its ATP levels go up. And the prediction was that maybe that would affect respiration. And indeed, actually, you see here the measurement of the respiration in astrocytes. It was not easy to do it in culture cells. We have to develop a new way to do it. But you see that it stops on its track when potassium is applied. And if you do this in slice, the normal decrease in oxygen concentration that is due to neuronal activity by high potassium. But if you wipe out the neuronal component like GTX, in response to potassium, you see an increase in oxygen. And this is because astrocytes are stopping their consumption of oxygen. So how we see it? We see that respiration in astrocytes is something that maybe we should look into it much more carefully than we have done before. This general simplification that astrocytes are glycolytic and neurons are oxidative might not be all the time. This is dynamic. And one possibility is, the way we see according to the data, is that oxygen is flowing into both cells. But when neurons become active, they signal astrocytes through potassium, they activate their glycolysis, and mitochondrial oxygen consumption in astrocyte is decreased. And then you have more oxygen for neurons. That would be the teleological interpretation of the data. So this could be an explanation of the presence of aerobic glycolysis in the brain, which has been quite mysterious. Why would you want to stop producing energy in a very efficient way when you need the energy? And this maybe is to do with oxygen. That is really the most precious short-time metabolite that there is. The half-time of oxygen in the brain is in the order of seconds, but lactate and glucose, they last for minutes. So oxygen is the real urgent thing in any brain. And this brings me to the last slide. So the hypothesis is still there very, very strongly, but obviously it's becoming more complex. And we have to add new players. Mitochondria in astrocytes is something that we should look into much more carefully. So how much of the oxygen consumption of the brain is in astrocytes and in neurons? According to some results, some people say that a third of the oxygen consumption of the brain is astrocytic, which is proportional to their volume. So this is important. And other signals like potassium, how glutamate and potassium interact, or with the other signals, ammonium and nitric oxide, is something that is interesting to explore. And of course, what neurons are doing. This is another world that we will leave you for another occasion. But there is, of course, research by us and other groups, which is very active, trying to understand really the intrinsic mechanisms in neurons, which are coupled to the mechanisms in astrocytes, mainly through lactate and maybe through other molecules too. And I want to thank collaborations of the last few years, and this is a recent, not so recent photograph in the lab when we could stand very close together. We don't do that any longer, of course. Well, thank you very much for your attention.
Thank you very much, Felipe, for that great talk. We can open it up to questions. I guess just like last time, people can unmute themselves and ask.
Can I ask a question? This is Pierre.
Sure.
Thank you, Felipe. Great, really great talk and really nice recent data. I think what you're showing with the, let's call it inhibition of oxygen utilization in astrocytes goes really along. I mean, it's too bad Mark cannot be here today because, I mean, he proposed this uncoupling many, many years ago, and also it was very controversial. And it was seen more like a sequence, you know, initial aerobic glycolysis in astrocytes. And the uncoupling, for those who don't know, is that during activation, there is an increase in glucose utilization in blood flow, but not so much in oxygen consumption, which is counterintuitive. And these are PET studies in humans published by Mark initially in the late 80s and confirmed many times, although some did not confirm, but I think most people confirm. So that means that during activation, somehow you increase glucose utilization without really using, at least for a certain time window, oxygen. And by the way, this was the basis for Seiji Ogawa to develop the fMRI. The idea with the BOLD method, blood oxygen level dependent signal, because paradoxically, and sometimes people forget this, the fMRI signal is due to the fact that there is more oxyhemoglobin compared to deoxyhemoglobin in the activated area, something that is counterintuitive. But what you show now at the cellular, almost molecular level, but certainly subcellular level, is extremely interesting because it's yet another evidence that there is a decrease in oxygen consumption, or at least not a commensurate increase during activation.
That's right. We always think about oxygen in terms of the vasculature, right? So you have demand, the vasculature is somehow activated, and you have more delivery. But the thing there is a complementary mechanism. So you have this redirectioning of oxygen from some cells to others, and it could be more complex. But if different kinds of neurons are involved, because of course, if we have this way to talk about the neuron and the astrocyte, which of course we well know is not true. In a bunch or in a voxel of tissue, you will have many different kind of cells. Some of them, maybe they are getting more oxygen, and some of them are restraining themselves in terms of oxygen usage, which would help the vasculature to deliver what is needed. We did some calculations. I remember looking at this story, that what would happen if all the neurons in a voxel would activate simultaneously. And that is totally impossible, even if the voxel is very small. That really kills the tissue. So yeah, we are very excited. Thank you.
Yeah.
Thank you very much. Are there any other questions for Felipe? I just have a quick question, Felipe. You know, these your sensors are beautiful and state of the art, and I was wondering if you anticipate any trouble similar to what calcium indicators give us in terms of buffering signals. For example, you have to interpret EGTA and BAPTA experiments differently. How are you seeing these results now in terms of their spatial temporal resolution?
Well, yeah, that's a very good question. That it depends on the sensor we're talking. So for the molecules which are present at high concentrations, like lactate, glucose, ATP, buffering is not an issue, because typical sensor concentrations are in the range of 10 micromolar. And these metabolites are hundreds of micromolar or millimolar. So if you do the calculations, it's not a major issue. It is a problem for NADH in the cytosol, particularly. In the cytosol, NADH could be nanomolar, and then fast events might suffer, and then you have this buffering that is maybe changing the intrinsic kinetics. But the good thing about metabolism is that it's very slow. So we're not expecting, but they might be there, but we're not expecting quick transients like in the millisecond range, like you see with the calcium. So even for NADH, or that might not be a big trouble. What is a real pity is that we haven't been able yet, I mean, the field of measuring NADH in mitochondria, even though the several groups have developed excellent sensors for the cytosol, somehow they don't seem to be working in mitochondria, which is a big mystery, and we need those sensors to work.
Great. Well, that's very reassuring that most of the, except for NADH, most of the signals are slow enough to measure accurately. Are there any other questions? So our next speaker is Professor Mariolina Linne, who joins us from the Faculty of Medicine and Health Technology at Tampere University in Finland. Mariolina is an engineer and neuroscientist with over 25 years of experience in electrophysiology and computational modeling of neural and brain signals. She leads the computational neuroscience group at Tampere. The focus of her research is on deciphering the roles of glial cells in synapses and neuronal circuits, as well as in brain signals recorded from humans. Mariolina will present to us her work on modeling astrocyte signaling and astrocyte-mediated long-term plasticity.
So good afternoon, everyone. My name is Marja-Leena Linne, and I'm going to tell you about modeling astrocyte signaling and astrocyte-mediated long-term plasticity. So as we've learned from Pierre and Felipe, there are many important roles for astrocytes in brain functions, and astrocytes are first of all involved in pre- and postnatal development, and they regulate the development of neural networks. They are involved in energy metabolism, which is the topic of this workshop, in homeostasis, survival, and also in many neurology, neuropsychiatric diseases. What is sort of less understood, but there is a lot of data accumulating now, is about the role of astrocytes in plasticity, learning, memory, and cognition. And this is also where I've been having an interest during the past few years, and where we've been doing modeling work to help explain the signaling that might be behind from the astrocyte perspective to these phenomena. So we know, as we also saw in the previous two talks, is that these brain functions are regulated by very complex astrocyte signaling. If we think about perhaps the main important homeostatic mechanisms that are there are this potassium buffering and glutamate sensing, which we already heard about a bit. And then in this illustration, we see that there are many other mechanisms that sort of link the astrocytes to synapses and then to the vascular system. And this is the discussion and topic that we are having in this this workshop as well. My main interest, as I said, has been on understanding the influence of astrocytes on the, let me take the cursor, on the synapses. But more recently, I've also started theoretical projects where we try to understand the neuronal astrocyte vascular interactions, especially explaining the fMRI signal and the sources behind fMRI signal. Many of this signaling in astrocyte is one way or another linked to calcium, and we are able to now measure these calcium signals in of course astrocyte soma, but also processes and end feed. And we know already that the signals in calcium signals in processes and end feeds, they differ from the ones in soma in terms of frequency, kinetics, and spatial spread. So already more than 3 decades, we've been measuring astrocyte calcium signals in vitro. And here is an example about cortical astrocytes at day 7 in culture. And this is a typical signal that you obtain. So there is this transient peak, and then there is this fluctuating signal. In this case, for example, for the serotonin 5-HT induced by the serotonin 5-HT. And in the very early phase of our modeling studies, we built up a model for such a case where you have this isolated astrocyte. So this work doesn't include synapses or anything. And we were exploring about the signaling mechanisms that are behind this signal. And with this model, we were able to show that the transient peak is due to the release of calcium from the ER. And then the delayed response is more due to the voltage-activated channels on the cell membrane of the astrocyte. We are also able to follow the spread of calcium signals with time in cultured astrocytes, as can be seen in this third illustration. The way we do computational modeling of biochemical signaling is the way that we do it for any other cell. And we basically consider either a well-mixed system where you model the ordinary differential equations, or then stochastic differential equations or chemical master equation. Or then you have this spatiotemporal system where you also take the diffusion into account and use the, for example, partial differential equations or Brownian dynamics. And there are many tools like STEPS, NeuroRD, EMSAL, that can be used to model biochemical signaling in cells. But we are starting to learn when we are doing this astrocyte modeling that there are challenges in using some of these earlier tools. And also new tools have been developed. For example, this Astro tool by Rusakov and Savchenko in London. So there is also a tool development going on now as we are kind of advancing with the modeling of astrocytes. So this is for those of you who have more biological background to remind about the modeling of astrocyte signaling. So like you model a signaling pathway or a reaction for any cell in the in a computational system. So you may have like 2 substrates interacting and you get a product. And such a system can be modeled by taking an ordinary differential equation, and you can follow the concentration changes of your system with time, as in this second equation. And if you go to a little bit more like a detailed system, for example, taking this ER in an astrocyte and have like 3 components, the leak current, the Cerca pump, and the IP3, where you get the calcium-induced calcium release from the ER. So you use such an equation where you have terms for all these 3 entities. So this is basically the methodology that we are using. In the course of this work, we've been also analyzing the previously developed models. And for those of you who are not so familiar with the modeling field, you should learn here that there are thousands of models for neurons, and you can see them, for example, in the model DP. But there are only a few hundreds of models for astrocytes. We analyzed 106 of them that one way or another have like a calcium component involved in these models. And we also mapped the origin of models, and you see here 4 different colors. So basically this illustration shows you on the right that there are 4 different types of models. So basically the models that are, for example, with blue color here, they are the same type. They use almost the same equations, maybe with a slightly different parameter values to explain some phenomena. And based on these studies, we concluded that the field of clear science and astrocyte modeling is in its very beginning phase. So then I turn to the second part of my talk, and there I will show you the work that we've been doing on modeling the astrocyte influence on synapses during development. We know that astrocytes have a fundamental role, for example, in activity-dependent synapse pruning, and there is fundamental work done by Kogla-Eroglu and many others. And particularly our long-term goal is to understand the dynamics of molecular signaling pathways and other mechanisms in cortical synapses when the morphology and activity changes during development. And as I said in the beginning, our special emphasis is on synaptic long-term plasticity. And for this particular work that I'm going to explain next, we made a hypothesis that astrocytes modulate long-term depression in developing somatosensory layer 4, layer 2, 3 synapses. And there is fundamental work done by Min and Nevian, and then also Banerjee and Paulsen on this topic. We all know that there are 2 forms of long-term plasticity, depression and potentiation. And when we experimentally induce long-term depression, we first induce a postsynaptic action potential that precedes the presynaptic potential, meaning that the timing difference between this protocol or this timing difference here is always negative. In the long-term potentiation, we do the opposite way. And many times this, when you do this pairing experiment, you call this spike timing dependent synaptic plasticity, STDP. This is a bidirectional change in synaptic strength, which depends on the temporal order and temporal difference of the pre and post-synaptic activity. And these are the typical curves that you obtain, and it's this LTD curve that we tried to mimic in the work of Min and Nevian. When modeling astrocyte neuron interactions, we of course have to also worry about the neuronal part, and we have already long track of record in modeling like the synaptic functions without the glial influences. And with our colleagues Janet Helgren, Kotaleski, and Avrama Blackwell, we did work on also analyzing postsynaptic signal transduction models for long-term potentiation and depression previously, and this has formed like the basis of our current work. So when we started understanding or trying to understand, when we were about to start to understand the role of possible role of astrocytes in synapses, we started from the work of Bean and Nevian published in 2012, where they made a really extensive, detailed study about the involvement of astrocytes in long-term plasticity in developing somatosensory cortex layer 4, layer 2, 3 synapse. So there, basically, you have a layer 4 pyramidal cell and layer 2, 3 pyramidal cell, and then the astrocyte, and the neuron that is sort of sending input to this system is the layer 4 spiny stellate cell. They did experiments, TLTD experiments, where they could see that there is a reduction in the synaptic strength, as in this figure. And they concluded in their experiments that this TLTD induction is dependent on endocannabinoid-dependent increases in astrocyte calcium signaling. They also did experiments where they show that this TLTD requires exocytosis of glutamate from astrocytes. And most importantly, blocking astrocyte calcium signaling completely inhibited this TLTD. So we use this study and also the studies from previous studies from Paulsen and Panerche and Rodriguez Moreno as a kind of starting point to understand what might be going on this layer 4, layer 2, 3 synapse during development. Linna and Nevian also looked at the astrocyte calcium signaling with 2-photon time-lapse imaging, as you see on the upper panel, and they were also visualizing the activity in astrocyte processes. Those processes that you see with green sort of start to have more activity during the pairing protocol. So clearly there is something going on, that some of the astrocyte processes have more activity during the pairing, and the others are sort of constantly active, like spontaneously active. So to make a story very short, I list here the 3 main assumptions that we did or put down to test our original hypothesis where the astrocytes are involved in TLTD. And we, first of all, our main assumption was that post-synaptically released endocannabinoid activates astrocytic endocannabinoid receptors and triggers calcium signaling in astrocytes in somatosensory cortex. And this came directly from the work of Bean and Nevian, but there were also others who have been showing it in other brain areas. Then the second assumption was that astrocytic calcium-dependent glutamate exocytosis, together with a spillover of glutamate from the synaptic left, has an effect on presynaptic glutamate release by modifying the release properties. There is a few references, major references listed here, but of course we know that there are also contradictory studies done in this field. In our model, we didn't model the glutamate release from astrocytes in the same way as in the neurons, meaning not in the way like vesicular way, but more like as a general exocytosis. And then the third assumption we made was that the link between the glutamate exocytosis from the astrocyte and the presynaptically released glutamate is the protein phosphatase calcineurin, which is activated by the influx of calcium through the presynaptic NMDA receptors. And there is several references that we use to obtain this assumption. There were also a few other assumptions that are listed in our publication recently published in PLOS Computational Biology, but these were really the main ones that sort of made us to build the model that you see now on the slide. This is a joint work. The model development is joint work with Tiina Manninen and Ausa Saudarkin, my colleagues from Tampere and Lithuania. And this is the overall schematic of the model we built for astrocyte neuron interactions during TLTD. So first of all, I should point out that this illustration only gives the main steps of information transfer in a synapse, and all the details are in the supplementary material of this paper. But to put it short, we have one compartmental presynaptic layer 4 spiny stellate cell, and we model all the major voltage-dependent ion channels and receptors on the presynaptic neuron membrane that are related to this phenomena according to our assumptions. Then the release of vesicles, and then also the calcineurin pathway that we developed ourselves for this model. So that didn't exist. Many of the components on the postsynaptic neuron, which is the layer 2-3 pyramidal cell, already existed. And there we model all major voltage-dependent ion channels, pumps, and most importantly, the AMPA, NMD, and metabotropic glutamate receptors. And then, of course, the source of calcium, internal source of calcium, the ER. And then this signaling pathway that leads to release of endocannabinoids. In the astrocyte, we model the endocannabinoid receptor and the calcium-induced calcium release from ER. Also, other mechanisms on the ER cell membrane, and the extracytotic release of glutamate that then binds to presynaptic NMDA receptors. So these were the main components that we took into account when we started exploring the possibility that whether astrocytes could be recruited in the phenomena that Min and Nevian presented in their 2012 paper. This gives you a glimpse of the methods that we used. Altogether, we used about 70 references to develop the model. First of all, the assumptions, and then the model components. There are some 80 differential equations, the types that are seen in this supplementary material. For example, in this case, the differential equation for calcineurin, and then about 170 parameters that define the model. And here are examples of some of the parameters, like the maximal parameter, maximal value for the calcineurin concentration. And there are a lot of such tables and tables for differential equations and parameter values in the appendix of this paper. So if you are interested in looking more details, you can go through. It took quite a long, I should say, to develop the model and to come up with all this, but it seems doable, I would say. So when we did the same type of pairing experiments, like in the experiments, we first introduced a protocol before TLTD induction, meaning we gave 5 pulses at 0.2 Hertz. Then we introduced the TLTD induction protocol, 100 post-pairings at 0.2 Hertz, and then the protocol afterwards. And what we are able to look at with such a system is that you see that we are able to, in all these different phases of this pairing protocol, we are able to follow all the variables that we cannot really measure simultaneously in vitro or in vivo. So we can measure, we can follow the development of cell membrane potential, calcium, release properties in the neuronal synapse, and then also the glutamate in the synapse. And here I would like to just point out the final figure here, that when we have introduced this TLTD protocol, we see a reduced synaptic glutamate release, as should be in this case when we are reproducing the TLTD. This slide pretty much summarizes the end result of our modeling work and validation. So with this model, we were able to reproduce the main result of Min and Nevian and also Banerjee 2014, where they see a decrease in the excitatory postsynaptic potentials. So the model, which is in black, quite nicely follows the experimental results. And the way one of the important things here is to realize that the way how we achieve this is that we introduce a delay in calcium in astrocytes through these complex signaling pathways that we have introduced to through the postsynaptic terminal, the astrocyte, and presynaptic terminal. So the crucial aspect is that you have to have a delay in the calcium activity in the system in order to end up with such a situation. And this shows the future potential of the models in silico modeling, where you can follow, as I said, all these variables of the model and see how they behave, and you can contrast them and validate against the experimental data. I should perhaps at this point out that we did validation of each of these terminals like separately first, and then once everything was integrated together, there was a final validation against this Min and Nevian work. And I should also point out that I would say that almost all, if not basically all of our parameter values should be in the range of biological range. So there is no that is completely out of scope of the So I conclude our modeling work that the fine sensitivity of TLTD to the temporal difference in a developing somatosensory cortex is achieved through complex molecular signaling. And this signaling that we incorporated is really indicated in separate experimental papers. We studied altogether 70 papers where we took the aspects of this molecular level signaling that led us to build this model and reproduce the results and make predictions about the dynamics of the molecular mechanisms, and then also how to link these together. And the second thing I would like to conclude is that during this TLTD, it seems to us that the single astrocyte could act as a delay factor for the fast neuronal activity. So in a way, like what this development of model taught me was that the sort of astrocytes help mediate integration of fast sensory processing with slow non-neuronal processing to modulate synaptic properties in the brain during development. And this is exactly what I said, like what I learned from this process myself was that it's very easy to say like that, well, some processing is like very slow and something is very fast, like we have tended to indicate, like for neurons and astrocytes. The other one is fast, the other one is slow, but it's really like, if you think about it as an information processing system, like they may have like both slow and fast may have their own implications for the end result. So it's not so simplistic that something is fast and slow, and then if it's slow, it doesn't matter at all. Well, a biologist probably knows it, but for an engineer, this kind of a model development, which is really detailed, was very fundamental learning experience as well. About the reproducibility, so we have the mathematical model available, fully available, and then also the code available in both GitHub and ModelDB. So if you are interested in trying this model or some aspects of it. And at the end, I would like to acknowledge my current research group and previous members, and then the funding both from the Academy of Finland and European Union and many colleagues that I keep interacting, collaborating. So thanks.
Thank you very much, Mariolina. We can open the discussion to questions from the audience now. Please unmute your mics and just ask a question. Felipe.
Thank you. That was a very, very, very nice talk, Mariolina. One question: when you say slow, are you talking about milliseconds, hundreds of milliseconds, or seconds? I'm asking this question, thinking in how to maybe in the next iteration to include metabolism in the loop, because you would expect that astrocyte will be doing things in the range of seconds during that loop in terms of metabolism.
Yeah, so it's a very good good point. So let me perhaps point out. So in the neuronal processing, I'm talking about milliseconds, of course, and in the astrocyte soma, I'm talking about seconds. But in the astrocyte processing, I'm talking about actually milliseconds or tens of milliseconds or hundreds of milliseconds. But still, there is some difference in the scales and with such a system, when you look at like instantaneous processing in synapses, of course, then maybe astrocytes don't like as such impose so many influences. But like when you look at these longer term influences, then definitely something like in the order of milliseconds, hundreds of milliseconds, and even seconds may influence. And I was also thinking like answering to your second part about the metabolic aspect and for example this lactate. So when I was listening to the first two talks, and of course I've been reading your work also before, but really I needed this kind of like talks to get my ideas about it, and I'm kind of now thinking that this lactate that you are introducing or you supply for the neurons might be like a precondition that is needed in order to, like perhaps during development, maybe also later on, to have, for example, this presynaptic NMDA receptor. So you need to have a certain balance there, and maybe the lactate is a signal for that. And then like when you go on and you need to learn something, like later on modulate something, like in our case with this endocannabinoid, perhaps to prune synapses during development. So then it's an additional step. So there may be many signaling molecules that it's kind of a cocktail that helps to modulate the synapse to what is the end goal, or what we are trying to do, or what the rodent is trying to do. So this is how I see. And of course, in our case, we only now model this endocannabinoid. Maybe it's kind of like it's probably important in this specific synapse during this developmental phase. But there, as I said, there may be many other signaling molecules. And yes, I would be very interested in also incorporating and modeling them.
Thank you.
Thank you.
Question?
Yeah, I have a quick question. I was interested by your work on endocannabinoid receptors, and you may be aware about the work of Juan Bolanos and other colleagues on mitochondrial cannabinoid receptors in astrocytes and their role in in learning or in social behavior. I mean, it's very another level of resolution, but clearly it has an impact on behavior. So I was just wondering if you consider these mitochondrial endocannabinoid receptors.
So we don't consider them yet, but I've been also recently studying this mitochondria. We don't incorporate the mitochondria like as such, like yet in these models, but I think it might be one of the next steps. And I'm very thankful for your presentations, because I actually got new ideas. So maybe I could use some of your data in the future to.
Sure, let's be in touch. That's the whole idea of the workshop.
Yes.
Yep.
Question?
Yes, Dan.
So yes, thank you for the nice talk, Mariolina. So over what kind of spatial domain do you think the astrocyte is integrating these signals? Is it at the level of a synapse or a group of synapses over the entirety of the astrocytic domain?
It's a good question. And I don't think we have an answer to it yet. So I'm kind of thinking that it's very specialized. So currently I think that it's like one process of an astrocyte takes care of basically one synapse, and so I'm thinking it's very specialized, but I think we need more and more evidence, both from the experimental work, but also like from the perspective that we really like model the full astrocyte and the full neuron. Like now we are only modeling one synapse and only one like one process or one small part of the astrocyte process. So we don't even have like good models for astrocytes so far. So we are lacking the description of voltage-dependent ion channels, for example, which behave, by the way, differently than in neurons. So we are really lacking. We are behind if we consider modeling the neurons. So we are not yet there to answer these questions, I think.
Felipe, did you have another question, or did you, your hand is...
Yes, it's a short one, maybe too technical, because Mariolina mentioned reaction diffusion and partial differential equations. So are you thinking about the astrocytic process moving in space, or they're just different compartments for the time being?
Yeah, so we have this fixed system. So we have only compartments, but in the future we should, in some of our other studies, we also use diffusion of molecules, and it would be very important in those cases where you have really few molecules like available or needed for some phenomena. But we can do both.
Great.
Thank you. Are there any other questions? All right, then we'll thank you very much, Mariolina. That was a fascinating talk and great questions as well. Our third speaker of this session is Professor Johanna Beyer, who is a research associate and lecturer in the Visual Computing Group at the School of Engineering and Applied Sciences at Harvard University. Before joining Harvard, she was a postdoctoral fellow at the Geometric Modeling and Scientific Visualization Center at King Abdullah University of Science and Technology, KAUST, our partners. She received a PhD in computer science from the Vienna University of Technology. Her research focuses on GPU-based volume rendering techniques for large-scale neuroscience and medical data. Welcome.
So thank you very much for the introduction. It's really an honor to be here. So today I want to talk about some recent works we did on creating interactive data visualizations for neuroscience, especially with a focus on large electron microscopy volumes and segmented volumes. So before I start, I just wanted to quickly introduce myself. Like you already heard, I grew up in Vienna, got my PhD at the University of Technology in Vienna in Austria, and then I did my postdoc at KAUST, where I worked with Markus Hadwiger at the Visual Computing Center. And after that, I moved to Boston to Harvard University, where I'm now a researcher. All right, so the Visual Computing Group, our main topics, I would say, are two different things. So on the one hand, it's visualization and computer graphics. And on the other hand, it's computer vision and machine learning. So we do collaborate a lot with the Harvard Center for Brain Science and with neuroscientists there. So on the one hand, we focus on ways for segmentation of large EM volumes and automatic synapse detection, things like motif detection. In my work, I personally focus much more on the visualization part and the visual analysis part of those large data sets. So I collaborate a lot with KAUST as well, and with Corrado and Turin about creating visual analysis tools that allow scientists to explore their data more easily. And this was a picture taken at one of the last solar eclipses, I believe. That's why we are all wearing funky sunglasses. All right, so before I talk more about the research we did, I want to very quickly start with motivating why we even need visualization, because I realize I'm the computer scientist here and most of you guys are biologists and neuroscientists. So a common definition of visualization is that data visualization is the use of visual representations to explore, make sense of, and communicate data. So examples of this would be looking at different variables and their correlation, or looking at like patient-specific data to plan detailed surgical interventions, or looking at the connectivity between objects in a network. So the main reason why visualization is so important is that it makes use of the human visual system and the power that it has in conveying information and kind of parsing information, in really gaining insight of the data. And another reason why it's so important is that it's particularly useful when there are no automatic or computational methods yet to completely analyze the data, where you really first need to look at the data to kind of figure out what is your hypothesis and what do you want to analyze next. All right, so in my talk today, I want to focus on 3 points in our research. And the first one is that in all our applications that we've built for neuroscientists, one thing we come across is that we need methods for computational scalability, because the data sets that we're dealing with are typically very large. So we kind of have to come up with interactive ways to display the data. And the second focus is that in addition to just computational scalability, we also need methods for perceptual scalability. And what I mean with that is that we kind of need visual abstractions to handle the data complexity, because even if you can display millions of synapses at the same time, that doesn't necessarily mean that you get more information out of it if you don't have a good way of how to kind of represent that information. And then throughout my talk, I will come back to different applications that we developed with our collaborators, with the domain scientists, based on the scalable methods that I talk about in the first 2 points. And then one thing I also want to make clear is that I will be focusing on this last step in the connectomics workflow. Like, I realized that going from the tissue, the imaging, registration, and segmentation is all a lot of work, right? And a lot of research is going into that. For the sake of this talk, I'm taking the easy way out, and I assume we have the labeled data, we have the EM volume, and structures have been labeled, and we want to focus on the analysis and visualization of that. All right, so let's look at scalable volume visualization. And we started this project because scientists at the Harvard Center for Brain Science approached us years ago that they kind of were collecting large amounts of EM data of brain tissue from mice and rats, and they really had no way of looking at it in 3D. So they wanted interactive rendering of terabyte-sized EM volumes. They also did not just have the raw data, but they also had a separate segmentation volume. So they wanted to be able to visualize, on the one hand, like sparse manual segmentation, but also dense automatic segmentation, where really every voxel in a data set is segmented and labeled. All right, so first, I would say let's start by looking at the data that they actually wanted to visualize. And those are some videos from our collaborators. So here you see a part of an EM image slice, right? And what they do is that they first collect this stack of slices to form a 3D volume. And then they do the segmentation. That means that in each slice of the data, they kind of define which pixels belong to a certain structure. And they do that for all the slices. So that then you can reconstruct like the labeled neuron in 3D. And here we actually see 2 neurons and their connection where they form a synapse. Just a quick question, is this video playing well for you, or is it as blocky as it is for me?
It's not really smooth.
All right, apologies for that. So it might be a poor internet connection. All right, so then if we zoom in on that part, we can see the different structures, right? But for now, you only saw the reconstruction of 2 of those structures. But what they're actually interested in is in the completely densely labeled volume. So what we get in the end is something that slowly, hopefully pops up on your screen like this, that you have this block that is so dense that you don't see anything anymore, right? So looking at this in 3D is kind of semi-helpful, because you just have so much occlusion that you still don't know what's going on. So that's another reason why we need clever ways for interactive visualization, for filtering and analysis tools. All right, and then if we zoom out from this data set a little bit to see the entire neuron, we would get something like this, right? But so here you see some segmented neurons that have been rendered with the cell body, but actually the part where we got the data from the previous video is just like this small area there, and that's already 1 terabyte in volume size. So keeping this in mind is that we really have to be able to handle large data. So we developed this scalable volume rendering system for this data. And I will spare you with the technical details for it because I think that's a little bit too detailed. But once we were able to interactively render these huge data sets, we were able to go 1 step further. And we built this tool called Connectome Explorer that then really focused on the interactive analysis of the connectomics data. So basically, we built it on top of the scalable volume renderer, right? But in addition to that, we also supported a more detailed exploration and analysis of the data by offering the possibility to ask domain-specific queries. And we had this query algebra where, just very quickly at this picture here at the bottom, you can see that we could ask a query where we say, okay, I'm interested in this red dendrite, show me all exons that are connected to it, but also only show me everything inside this region of interest to get to the final result. So it was kind of a more intuitive way to allow neuroscientists to kind of analyze and explore their data that would be too big to just look at the entire thing. All right, so I would say this was the part I wanted to talk about that focused on computational scalability. But like I mentioned before, visualizing volumes of this size also requires us to think about the way, how much detail we want to show. And that's the second focus we do a lot is visual abstractions to handle data complexity. So one project in this area I briefly want to talk about is Abstractocyte. So it's the combination of astrocyte with abstract visualization. And in this project, this we did together with KAUST and Corrado. In this project, they were interested in the analysis of astrocytes in brain tissue. And we had high resolution and segmented EM data. And the goal was to explore, on the one hand, the morphology of astrocytes, but also look at proximity patterns. So look at different structures in relation to astrocytes, for example, the distribution of mitochondria in an astrocytes or the location of synapses. And the third goal was to also analyze glycogen distribution patterns and the relationship between glycogen, mitochondria, and synapse activity. So the main challenge here were that first, the morphology of astrocytes is very complicated. So as astrocytes kind of surround neurons, right? Because there are those supportive cells. And that means that right now here in this rendering, you see 2 neurons, but if we add a single astrocyte to the visualization, this is what you get. So suddenly you realize that you really need to be able to show a simplified version of an astrocyte somehow that does not completely clutter the view. And the second challenge was that the complex morphology of astrocytes also makes the analysis of certain distribution patterns more challenging. So here we see the astrocyte in red and glycogen granules in green. Here's actually a zoomed in version of that. And the neuroscientists were interested in questions like, is there a correlation between the glycogen granules and whatever synapse strengths or something like that? But for that, we also need to know the proximity and location of nearby synapses and structures. So to answer all these questions, ideally different visualizations or different visual abstractions are needed, where really for one certain task, you want one visualization, for another task, you might want another visualization. So depending on the task, we created a system where scientists can choose their appropriate abstraction level. So we came up with different abstractions for astrocytes as well as neurites, going from the concrete on the left, which would be surface rendering, to non-photorealistic rendering, to a representation that only shows like their skeletons or their simplified and straightened skeletons, up to the most abstract version, which would be on the right, which would be a node link diagram or heat map shown over a node link diagram. And the most important part of this project was to offer users an easy and intuitive way to navigate between those abstraction levels. So we designed this 2D visual abstraction space where users can interactively navigate this space, and the visualization continuously changes and interpolates between the different abstraction modes. And by doing this, we kind of preserve the visual context, right? Because it doesn't just switch from one view to another. You get the slow interpolation and you can follow what is changing and what is happening. So in this abstraction panel, the bottom left is the origin, which is the most concrete visualization, and the top right is the most abstract one. So let me start this video. So when we move along the y-axis, or the user moves along the y-axis, the astrocytes gets abstracted from mesh to skeleton to color coding on the surface of nearby neurons. So in this example, we actually still show the glycogen granules. And then if we move along the x-axis, we change the neuron representation. So we abstract the neuron from mesh to skeleton. And here we still show the skeleton of the astrocyte. And then when the user navigates to the top right of the abstraction panel, we actually go from a 3D view to a 2D representation, where we use a force-directed layout to show neurons with a heat map showing astrocyte proximity. So our collaborators use this for evaluating different use cases. So one main aspect for scientists was also to kind of use this for visual proximity analysis. So visually displaying the proximity of a specific cell structure A to a cell structure B. And in 3D, we can do that by mapping a distance field onto the surface. So you can see like highlights of red where the astrocyte is close. In a 2D, we can use a heat map on top of the network diagram, something like that, to kind of show proximity of astrocytes to neurons. And the second use case, what they wanted to do is to do some initial glycogen analysis, because glycogen tends to appear in clusters in different locations inside the astrocyte. So we use first a clustering algorithm to compute those clusters. And then we implemented different visualization modes. So in our framework, we support 4 different visual states that would help to study the glycogen. So on the left column here, the left 2 pictures, you can see a heat map in 3D and in 2D, but we can also color code clusters and color code object based on mapped clusters. So this is, I know this is very quick and this is just meant to be an overview, but this is basically a way how we used visual abstraction and really focusing on what the user task is to kind of create and design the visualization that's most useful for that. All right, and I actually want to talk about one more project that is still very much work in progress, where we focused on the visual analysis of spatial neighborhoods and comparison. So the idea in this project is to focus really on neighborhoods and how to analyze, quantify, and explore them. And it's still an ongoing project, so I'm only showing some preliminary results here. So the 3 main goals of the project is, on the one hand, we want to look at spatial neighborhoods in connectomics data, so in EM data. And we want to analyze spatial distribution, also look at distances between structures and 3D morphology. And on the other hand, to do that, we kind of need a scalable visualization or visual comparison framework that allows us to compare a wide number of elements and also be able to compare, for example, 2 structures, but also to compare hundreds of structures. So we kind of need to be flexible in what our visualization supports there. And also what we wanted to do is, on the one hand, we wanted to create this general framework for neuroscience, but we also want to allow individual people to kind of customize it for their distinct use case. So one of our use cases that we worked with Corrado is mitochondria analysis, and kind of focusing the neighborhood analysis and the comparison on mitochondria analysis. So here are some of the detailed domain questions that we're trying to support in our tool. So looking at or analyzing how synapses are distributed around a mitochondrion, or how mitochondria is positioned within a nerve cell with respect to the cell boundary. Does it get close to the boundary or not? And also with respect to spines. So here's our approach. And the idea is that first, users can specify basically an analysis scenario that they're interested in. So for example, analyzing mitochondria. And usually a scenario consists of several subtasks that they want to do, like the ones that I just talked about for mitochondria analysis. And then based on this basically initial configuration, during the actual visual exploration, our tool can automatically adjust its user interface and kind of suggest visualizations that best help the user in their task. So basically, really customizing the experience for the current user task. So one example would be that if the user wants to look at spine coverage for mitochondria, we would show a visualization that's really optimized to show that more than just looking at the 3D rendering. Another example would be that if the user is trying to compare different neighborhoods, that depending on how many structures or how many neighborhoods they want to compare, that we can automatically adjust which visualization we show. Because if you only compare 2 structures, you can really compare them in very much high detail and use your screen space to show all the details. But if you want to compare hundreds of structures, then you kind of need an overview or a rougher type of comparison that fits onto your screen. All right, so yeah, basically you specify your use case first, and then we automatically adjust the visualizations that are shown based on the current task to kind of help and guide the user in their analysis. So here's the workflow in a little bit more detail. So basically what we show here is that on the left, the user starts with this configuration file where they kind of specify their analysis or scenario. And then during the analysis, on the one hand, they can look at individual neighborhoods in 3D. So that's what you can see in the bottom row here, like local neighborhoods around mitochondria that show the mitochondria, the surrounding cell, and nearby synapses. And on the other hand, we also offer more quantitative view for more quantitative analysis that you can see here at the top that kind of support the comparison of those structures. And we always have designed like different views for different cardinalities. So basically we show a different comparative view for if you compare 2 structures versus 10 versus 100. All right, so next I'm going to show you a screenshot of the actual system because I think that will make things a little less abstract. So here's the entire user interface that we currently have. On the left here, you see basically the mitochondria selection panel. So users can go in and look at select the mitochondria they're interested in. On the right here are the visualization parameters where we show for each of the 3 tasks, like different visualizations that we suggest that the user uses to analyze their data. And in the middle is the actual visualizations. So the bottom shows the 3D views of the selected neighborhoods. And in this example, the top view shows a violin plot, where for each mitochondrion, you get your own plot, your own violin plot. And the Y position represents the distance to the cell boundary. So the higher on the Y-axis, the further away it is from the boundary. And the width of the violin plot shows how much of the mitochondrial surface has that exact distance. So by kind of looking at the violin plot, you get a quick overview of how close is this mitochondria to the cell boundary. And this is just one task. Like I said, depending on which task the user is interested in, we can show different analysis views. All right, I know this was a very quick tour of some different projects we did in visualization for neuroscience data. I would say some key takeaways for connectomics visualization is that the main challenge is scalability. So on the one hand, that means computational scalability, being able to handle the amount of data that we get. On the other hand, it's more limiting the cognitive load that you put on the user, coming up with nifty or good visual encodings that kind of abstract the data to make it easier to really analyze it the way that you guys want to analyze it. Another point is that we really need quantitative methods for visual analysis, going one step further from just exploring the data in 3D, being able to like look at numbers or look at more quantitative views to compare and to measure things. So those are the next steps that are really important. And another thing that I really want to stress is that really the domain goals is what inform our visualization solution. So what I mean with that is that when creating the visual abstractions, for example, it's really very important to closely collaborate between computer science and biologists or neuroscientists to kind of make sure that the abstractions make sense from the biology side, right? That it's not just visually pleasing, but that it actually makes sense. So usually we have a lot of iteration steps going back and forth to make sure that we get those abstractions right. And even though there's still many open issues and challenges to be tackled, data sets are still getting bigger. I would say that by this close collaboration, we can tackle those challenges. All right, so that concludes my talk. I quickly want to thank my collaborators over the years, because without them, all this work would not have been possible. And with that, thank you very much.
Thank you very much, Johanna. You've developed some incredibly beautiful techniques and corresponding images. Very fascinating. I'll start with one question. So your proximity analysis is potentially really very useful. Have you noticed any correlation between astrocytic proximities to synapses and glycogen content in astrocytic processes?
Right, so I would say right now we are still kind of in the testing phase of this, like since it's, so especially this last project I showed, we'll be able to give like really quantitative measurements back. So I wouldn't want to talk about like biological findings, and maybe refer that to Corrado if he's available.
All right, well,
yes, I'm here. Yeah, so I think to answer this question, we should think back to Abstractocyte, which was really the tool that that has been developed and thought about the analysis, thinking about the analysis of glycogen specifically. So we have seen some correlations, which is not shocking, because in any case we know that, or we can imagine that to a certain extent, the presence of glycogen, in order to be exerted, must or should, in principle, somehow be related to the presence of glycogen. Now, saying that, we should still be quantifying this in a more solid way, I would say, and this is something that I will briefly talk about tomorrow. And this is part of the work that is being carried on by Marja Veloz, who is our PhD student, who recently started together with Pierre. But the answer is yes. I mean, we looked specifically for this correlation, and yes, we found these correlations.
Okay, great. Well, thank you very much. From the field of general participants, I'll take more questions. Johanna will take more questions.
I would have a question. This is Felix.
Okay, Felix.
Thank you, Johanna, for this great talk. It's really exciting. How much of this relies on the fact that you really segmented the tissue before, and if it does, I mean, could you think of sort of doing it on the fly? I mean, in the sense, because when one looks at such a small area when you when you do this, right? I mean, you're in, and sort of the question whether you really find what you want in that region, or whether you need to look next door. So what are your thoughts about kind of combining that with sort of some of the segmentation efforts?
Absolutely, very good question. So in the Abstractocyte project that I showed, and also the comparative neighborhood analysis, we do rely on having segmentation information. So to really be able to just visualize the one structure you're interested in, right? You kind of have to know these voxels belong to that structure. We do have general visualizations for just looking at the EM data, like that I very quickly talked about the volume visualization framework, where you can show you can look at the entire data. But for more detailed analysis, I would say you would need some kind of segmentation. One way to approach this could be to be able to compute some things on the fly, like you mentioned. Typically, I would say that EM data is very complex and hard to segment. To get like a good high quality segmentation out of it, that's pretty tricky to do on the fly. So usually I would say we would expect to have pre-segmented volumes or areas. One other field of research that we're looking into is to kind of have automatic or semi-automatic interactive proofreading, where we start with a segmentation that might not be super correct, and then have some visual tool that kind of allows users to quickly correct that segmentation to make sure it's right. That's more integrated into their workflow than it currently is. So you could think of some ways to combine, like looking at the data and then specifying, I only want to segment this area in more detail, and kind of doing that more on the fly before moving on. But currently that's not really in our system.
That makes sense. It just sort of reminds me a little bit of this elastic. I mean, you annotate a few things, and then you kind of say try, and sort of to kind of combine these two things. But what you're saying makes perfect sense. Thank you.
I'll just ask you one more question, Johanna. What developmental stage are you focusing on right now for collecting all of this data?
You mean for tissue samples?
Yes.
I will refer that to Corrado, maybe.
Data collection, especially for EM, for this kind of technique, is always quite tricky, and strictly depends on the machine that you're using, on the resolution. But typically, for a volume of about 100 cubic microns at high resolution, you might see something between 1 and 2 weeks acquisition time.
Actually, my question was, what developmental time point are you focusing on for data?
Oh, sorry. I totally mistake your question. So for Abstractocyte, the data set that has been shown was from 4-month-old and 24-month-old mice. So from young adults to rather old, and these data came from a paper that we published with Graham Knott on PLOS ONE. But then we have been working on also with other data sets from adult rats, from 2 months old rats. And then we have plans of working on another data set, which was a progress in neurobiology data sets. That's a P14 rat. Yes.
Great, thank you very much for that answer. Thank you very much, Johanna. That was a scintillating talk. Fantastically beautiful images and amazing tools.